Good afternoon, everybody. I'm Al Somer, and I have the distinct pleasure of chairing the Vision Prize jury for the Champalimaud Foundation. Lenore Beleza, president of the foundation, regrets that she's unable to join us this year, but asks that I convey her continuing appreciation of ARVO's interest and support for this important annual event. As many of you know, the foundation was established by the bequest of Antonio Champalimaud in memory of his parents. This past year, on the centenary of the Portuguese Republic, the foundation dedicated its new, breathtakingly beautiful, fully equipped research and clinical facility, the Center for the Unknown, located precisely where earlier Portuguese investigators of a very different sort, those bold navigators of the 15th and 16th centuries, first set sail across uncharted waters to discover other unknown lands. The new institute is focused on neuroscience and cancer research, but one of the foundation's first investments 5 years ago was the establishment of the Champalimaud Vision Award. This award recognizes fundamental discoveries in vision research and on alternate years, innovative service programs for the prevention of blindness. Both awards carry a stipend of €1 million, which is meant to assist the recipient in furthering the work for which they are being recognized. The 2010 award, which we celebrate today, was bestowed on the recipients at a special ceremony in Lisbon that was presided over by Portugal's president and attended by its prime minister. Several of my fellow jurors are here today. I'd like to ask Paul Sieving, Nag Rao, and Jose Cunavas to please stand and be recognized. They're up here. I'd also like to thank ARVO's trustees and leadership for making these ARVO Champalimaud lectures a regular and distinguished part of the ARVO annual meeting. This is the second year that the award honors outstanding vision research. The jury carefully and with great difficulty weighed the merits of 37 outstanding nominations. As you already know, this year's honorees are Tony Movshan, director of NYU's Center for Neural Studies, and Bill Newsom, a Howard Hughes investigator and professor of neurobiology at Stanford. They are close colleagues and longtime friends. Working together and sometimes separately, they have unified the fields of visual perception at the behavioral level and visual information processing at the electrophysiological level, creating a single, seamless science of seeing. They causally link the response properties of neurons in the visual cortex to perception and action, and in so doing showed how computations by neural circuits can account for the way we see the world. Tony will present his presentation first, to be followed by Bill. Tony?
Thank you, Al. Thank you, members of the jury, those of you kind enough to remain here and skip the beach to be here. And thanks, of course, most of all to the Champalimaud Foundation for what is a tremendous honor and an enormous privilege. I can't emphasize enough to all of you, first of all, how fabulous the Champalimaud Center in Lisbon is, and you should definitely go and get a look at it if you have a chance. And secondly, how important to me and to Bill and to our colleagues this prize is in recognizing our work. It is work that we shared and we did with many others, and I don't think that it is really even reasonable for the two of us to share it, because it is in fact work by many, many people, some of whose names you'll see going by on my slides or Bill's. It's, I think, particularly apt that we're giving talks here because although we don't usually confess these things in public, ARVO is actually where Bill and I first met and where we formed our first collaboration over a long and very contentious dinner, which consisted mostly of Bill saying I had to be wrong and me saying actually no, I wasn't. But broadly speaking, that's how we've gone on for the last 30-odd years. This is also the meeting at which some of the work for which we were recognized was first presented. So we met at the 1981 meeting our work was presented together at 1989 meetings and at other meetings. And so it's nice to be back here and particularly welcome and particularly fitting, I think. Now, let me now go on and talk a little bit about the topic I've chosen, which is up on the board here, Encoding and Decoding of Visual Information by Cortical Neurons. There's not enough time, you will all be glad to hear, for me to give you the kind of account that I would enjoy giving of the work that Bill and I began and that many of us have worked on. But I hope to give you sort of a bit of a flavor for the kinds of questions that we've addressed and the ways in which we've tried to tackle them. My job, as it often is to be a straight man for Bill, who will come second and who will take advantage of what I've said and correct the errors and enhance the truths. And hopefully at the end of the day you'll have something to take away in addition to the drinks that I hope you will share. So the structure in which we work and the structure with which we are concerned is the macaque-monkey cerebral cortex, which is often represented as here in this flattened representation, which was first done by David Van Essen and his colleagues in the late 80s and early 90s. The visual areas of the brain are all shown here in color, and they are, as many of you know, richly interconnected with one another, so that you can draw a diagram of this kind which connects the different visual areas. This diagram is a little different from the one you're used to seeing because the areas are represented by boxes proportional to their actual size, and the bands connecting them are based on estimates of how many fibers actually connect them. The small red area in the center, called MT, is the one in which Bill and I, in effect, first joined forces. And it is an area which is very predominantly associated with visual motion processing. The general task of neuroscience that we often try to frame in terms of dealing with questions about systems has to do with relating the anatomical map, now minimized at the bottom left of this slide, with the sort of cognitive psychological map that's shown to the top right, which I stole from a paper by Ted Adelson and Jim Bergen. And the idea that many of us have carried around is that particular modules in early vision, by early vision we mean sort of early areas of visual cortex, process particular kinds of information and then pass that on to higher centers. And what I'm going to tell you about in the next few minutes is a little bit about how one of those boxes, the motion box, acts to encode visual information, and then a little bit about how some of that more processing that's referred to in those highly specific later stages of that diagram actually go on to deal with some of that information. Now, the big hint about motion processing comes from the fact that cells in the brain are sometimes directionally selective for one direction of visual motion over another. And so these data are from Hubel and Wiesel's study of primary visual cortex in monkey done in 1968. And if you look at the responses of the cells here, I'm not going to point my green pointer at one screen and to the exclusion of the others, but in the middle trace you see a burst of spikes associated with motion up and to the right, and a much weaker burst associated with motion down and to the left. But in primary visual cortex, only a minority of cells have this property, whereas in MT, the area that actually Bill introduced me to, most cells have a tuning curve for direction of motion like the one shown at the bottom right. The polar plot shows the response of the cell as a function of the angle of the motion of the target. And we became interested in the question of how the signals from these direction-selective cells might support the perception of visual motion, and in particular the perception of the direction of visual motion. Now we have a sort of semi-unfolded view of the macaque's brain to the top right, and down on the bottom left is a cartoon of the stimulus that we used to probe this. And the stimulus choice is worth taking a moment to consider, because when you do experiments of this kind, to study, for example, one particular visual process like motion, as many of you know, you can't just assume that because a moving object is crossing the screen that motion-sensing mechanisms are the ones that are used to process it. So the stimuli we used are what are called random dot kinematograms, and they consist of a series of random dots. They always have the same number of dots, and what they vary in, as you can see at the bottom left of the screen here, is the fraction of the dots that move coherently in a single direction. And what I'm going to do in the next few slides is ask all of you to serve as monkeys for the purposes of doing a psychophysical task. In each frame, what I'm going to do is I'm going to show you a fixation point, which will be a yellow dot. And then below and to the left of the fixation point will be a field of moving dots, and you're supposed to shout out as soon as you can see which direction the dots are moving. You ready? Very good. Okay. That's, of course, at 100% coherence. Come on. You can do better than that, all right? We're, of course, going down in coherence. Okay. No laughing over there. I don't know what you guys are doing. No juice. You just get the joy of a job well done. Keep going, okay? Okay, I'm not hearing a lot of precise responding here. Here? Okay. And finally, one more of those. Williams, you got that right. Okay. So what you saw was a series of trials in what you might imagine to be a psychometric experiment asking how performance varied with dot coherence. And what you observed was two things. First of all, obviously, as the dot coherence reduced, you became less and less good at judging the direction. And secondly, it took longer and longer to do that judgment. And one of the things that makes this task intriguing is that it requires you to integrate information over time and over space in order to perform accurately. When all the dots move in the same direction, of course, you can effortlessly get it right almost immediately. But as soon as you introduce the noise dots, what you have to do is, in effect, average over space and time to get the answer right. And it turns out that cells in MT behave in a way that suggests that they do something similar. So these are data that show that, for example, neurons from experiments that we did respond in a way that depends on motion coherence. In each plot, the solid symbols are responses to dots that move in the preferred direction for each neuron. The open symbols are for dots that move in the non-preferred direction. And as the coherence increases along the x-axis, the separation between the curves grows. Now, in thinking about this in psychophysical terms, it seems intuitively reasonable to think that when cells preferring right respond reliably more than cells preferring left, you'll be able to do the task. And when cells preferring right don't, then you'll be doing it poorly. And if you look at these curves, for example, the one on the top right, you'll see that at 5 or 10% coherence, the responses are only slightly different on average. But it's not going to be the average that actually matters. What's going to matter is the statistical distribution of the signals in these different classes of response, and I'll show you a little bit about why that is. So the experiment that actually began all this was done in a way that's diagrammed here. The monkey fixated the red fixation target to the bottom right of the upper panel. The receptive field of the cell in MT was isolated, and the dot field was placed on top of it, and the motion of the dots was arranged so that it directed either in the preferred or the anti-preferred direction for the neuron under study. And the time course of a trial is shown at the bottom. The essence of it is that at the end of a forced period in which the monkey had to watch the dots move, he made a choice to either one of two targets which indicated which direction he perceived. And at the same time, what we could do is use a statistical method to ask how reliably the cells differentiated responses to the two directions, allowing us to make a comparison, shown here, between what we call the psychometric functions and the neurometric functions for the discrimination of motion direction. Coherence is the x-axis in all these plots. The filled symbols show data taken from the neuronal data. The open symbols show results taken from the behavioral data collected on the same trials. The result on average suggests that neuronal and behavioral performance are very similar. And if you take a measure of the performance, just as, for example, the threshold coherence required to evoke responses that are accurate 80% of the time, and you plot the ratio of the neuronal to the behavioral thresholds over many trials, the answer is what you see on the right, a distribution whose modal value is very close to 1. Now, this was actually a very interesting and an intriguing result, and it excited us quite a bit, but it is a result which, as I hope most of you realize, can't possibly be true in the sense that what it implies can't possibly be right. It implies that in some way the monkey pays attention to the neuron that we happen to be recording in order to make the judgments that we happen to be measuring on a particular set of trials. The monkey, however, does not know which neuron we're recording. He has 4 or 5 million neurons in MT, and therefore his challenge is to deal with the signals carried by many other neurons and make his mind up on the basis of those signals together. The fact that the ratio of thresholds is close to 1, however, shows that neurons in MT actually carry information which is reliable enough to use for this judgment and suggests that these neurons might be directly associated with perceptual decisions. And it turns out that there's another aspect of the data that shows another piece of evidence in favor of that proposition. What's shown here are two distributions. The upper one is a distribution of spike counts on a set of trials for a particular visual stimulus which actually had on average no net motion, 0% coherence. The upper histogram shows the response distribution on trials in which the monkey chose the direction that the neuron we were recording from preferred. And the lower distribution, which is different in its mean, as you can see, shows the responses on average on trials where the monkey chose the other direction, the anti-preferred direction. Now, the fact that these two distributions are different suggests that there's information in the responses of these sensory neurons in MT about the decision that the monkey will make at the end of the trial, after the dots go off and he chooses to program an eye movement. And we became very interested in the question of how that information was carried by this population of neurons and what it meant for downstream processing. A way that we can actually capture what's going on here is to ask in a statistical sense, if we only had the information from this one neuron, how reliably would we be able to tell which decision the monkey would make on any given trial? And we call that quantity choice probability, and we plot over here a distribution of the choice probabilities for a particular set of neurons, all on these trials where there was no net motion. So the thing to remember here is that there's no information from the stimulus trial to trial about what's going on here. All that you're looking at here is, in effect, an associated response related to the variability of firing that cortical cells have from trial to trial. And the fact that this distribution is shifted to the right and has a mean which is highly significantly, although modestly above 0.5, means that the population carries a good deal of information about the impending decision well in advance of the actual action. Now, there are a number of different ways in which this can be interpreted, and I don't have time to dwell on them. It is an interesting finding which is still generating quite a lot of active research. But let me give you an intuition for the way in which we initially thought about it, and to be honest, the way in which I still think about it, arising from neuronal responses. Suppose we have a pool of neuronal responses, and what I have to ask you to imagine is the responses of two neurons, one of which prefers, let's say, up, we'll call that the preferred direction, and another which prefers the opposite direction, down, we'll call that the anti-preferred direction. And you can see them on the ordinate and the abscissa of this plot. The diagonal shows the line of equality, and what we're going to do is treat a very simple model of decision in which we say, if the preferred direction neuron fires more, we'll choose preferred. And if the anti-preferred direction fires more, we'll choose anti-preferred. So we can divide all these trials into those in which the anti-preferred response is larger, and we therefore choose anti-preferred. And the preferred, and those are in blue, and then the other trials are in red, where the monkey presumably would choose preferred. Now, the key thing, the intuition I want you to get here, is that if you are measuring the response of the preferred neuron, which is what you can see along the x-axis of this plot, and you compare the responses on these two kinds of trials, the conditionalization of the response distribution will cause the average activity to be different. And in fact, if I compute those distributions, as shown here, what you see is that the responses on the trials in which the monkey said preferred, are larger than the responses on the trials where the monkey said anti-preferred. So conditionalizing the responses, which are purely varying as a result of noise, based on the monkey's decision, shows this difference. But what's key about this is that it requires that these neurons be associated with the preferred, non-preferred decision that the monkey makes. Now, this particular case, the idealized two-neuron case, has a choice probability associated with it that's quite high, about 0.85. And I remind you here that the actual observed values were much lower than that, suggesting that the monkey was using more than two neurons to make up his mind. And the sort of framework in which we think about these decisions is kind of captured by this cartoon, in which the left-hand boxes contain pools of neurons, numerous neurons, all of which signal different directions of motion, but in which we take the upper box to represent all of those neurons that have an upward component to their response, and we pool them to make the upward signal. The bottom box shows those neurons that have a downward signal, and the decision is simply based on which of these two neuronal pools has more activity. Now, in this framework, the question of how activity of single neurons is associated with decisions is very much akin to a political decision, all right? And I would invite you all to consider yourselves to be neurons voting for either up or down. Now, as you well know from recent presidential elections and other exciting things in American politics, your individual influence on an election is not discernible. If there are 100 million voters, your particular vote isn't going to have any impact, and therefore your choice probability, if you will be basically 0.5 or 0.5000001, based on the fact that there are 100 million other voters who are voting at the same time. What makes your vote appear to have an impact is your correlation with other voters, for example, voters in your political party. And if your other voters are correlated with you, then you will have a choice probability. And that's a key intuition here also. Neurons can have a choice probability only if their activity is correlated with that of other neurons with similar preferences, which contribute similarly to the decision. Fortunately, Udi Zohari, working in Bill's lab, showed some years ago that neurons do have that correlation. This somewhat messy plot shows that the cluster of points to the left in particular shows that if you record two neurons near one another, and you compare their activity across many trials, that activity is correlated with a sort of standard correlation coefficient value R of about 0.2, but that is prominent only when the neurons share a preferred direction. It's not evident when they are preferring other directions. And so this poses now a question for us. We have a hint from choice probability measurements that these neurons are involved not only in capturing motion data and encoding it, but also in their information is being used to decode signals. And so what I'm going to do now is switch gears and talk briefly about another set of problems that have interested me more recently, which are the problems of how you decode the activity of populations of neurons like those in MT. So to start with, we have these notions of encoding captured here by these cartoons of direction selectivity and coherence-dependent responses in MT cells. And what we now want to turn to is the question of how we decode these responses for the control of behavior. The noisy Gaussian-looking plot on the right-hand side here is not something you should look at as I would have typically looked at it, as the tuning curve of a single neuron. What you're going to look at that with, the eyes you're going to look at that with for the rest of the talk, will see each of the arrows underneath it representing an individual group of neurons preferring the indicated direction. So what you're looking at now is a population response, showing the response of a population of MT neurons in response to a single target. And your job as an observer will be to work out what that target actually was. In this case, it should be fairly clear that the target was some direction close to vertical. But in other cases, for example, where the coherence is low, it may not be so easy. And the histograms down below are simply to remind me, to remind you, that the choice probability data indicates that there is, in fact, an association between this population response and the judgments that observers make. So the problem we're now faced with is of evaluating sensory evidence. And the way most of you in the room, I think, who have experience in psychophysics would probably want to think about this problem is to ask it in this way, to pose it in the sense that if you're an observer inside the head and all you have is the activity profile of populations of neurons that are connected to the outside world, you have a statistical decision to make, which is to evaluate what the most likely stimulus or stimuli in the world were that could have given rise to the activity you observe. And this is the sort of computation of likelihood from neuronal data. Now, the idea that neuronal populations could represent the likelihood of stimuli is one that goes back about 20 years. It came up in sensory work by Peter Foldiak and actually in motor system work by Terry Sanger. And they both had very much the same intuition, and I'm going to try and walk you through it quickly here. The idea is as follows. Suppose we have a neuron whose tuning curve is shown on the top left. It responds in a way that reliably varies with some parameter theta. And suppose that we measure the response of this neuron to two different values of theta, theta 1 and theta 2, the one associated with the brown and the green bands. The gray band is the variability of the neuron. And down below, we have actual distributions of expected responses of the neuron to two different values of theta, theta 1 and theta 2, the green one in the middle representing the theta value to the right, and the brown one at the bottom representing the theta value to its left. Now, under those circumstances, what you can see is that for a particular measured value of the response, which is the response N1, and I'm going to have to actually point this out because this response N1, what you can do is you can read these distributions to ask what is the probability that that particular response would have been observed on the basis of the tuning curve of this neuron. And the answer is you can read it off each of these distributions. So now you can take a second neuron, which has a different tuning curve, and these are the response distributions associated with its tuning curve, and you measure a different response from that neuron, we'll call it N2 spikes, and that gives you another estimate of the probability. Now, these measures can be taken as the probability that spike count N1 was associated with stimulus theta 1, or the probability that spike count N2 was associated with stimulus theta 2. And since these are conditional probabilities, we can multiply them out across an arbitrarily large population of neurons to learn what the likelihood is of any particular value of theta. Now, the problem is that you can do this statistically, and you can see it in a graphical form like this, but the idea of doing it computationally in neural circuits was one that several people kind of beat their heads on for a few years after these papers came out. And I had the great good fortune to have a very talented student, Mehrdad Jazayeri, who worked out how to do it. And the answer turns out to be extremely simple. This particular diagram may not look extremely simple, but it is, because all it is a network which takes the signals N1, N2, and so on from a bunch of sensory neurons, weights them appropriately, and adds them up to compute a function which Mehrdad showed was actually proportional to the logarithm of the likelihood that any particular stimulus was present in the environment. This is a strategy for recoding sensory data in a form that allows likelihood to be measured from it. And There's a particular version of the model that you can use to study the behavior of cells in MT. This shows a cartoon of it. This is a population response for a group of cells in MT, as I indicated before. This is a feed-forward weighting of a simple model to compute the likelihood that the particular direction here shown as leftward was actually present. What this curve means is that in order to do that computation, you take cells in MT whose direction preference is near the direction you're trying to estimate, and you count them positively towards the likelihood. And you take directions about 180 degrees away, and you count them negatively. This is very much like the idea that you take the difference between two populations of neurons preferring opposite directions in order to compute the outcome. And so you can do this, of course, for other directions of motion, this one or that one, and what you end up with is a cartoon which suggests that you can estimate likelihood with populations of MT neurons by having multiple stages of this recombination of inputs. The intuition here that you need to carry around is that you take individual sensory neurons and you pool them using these optimally designed weighting functions so as to basically improve your estimate of the actual existing conditions in the world that gave rise to the distribution of responses that you measure. Now, MT is perfectly nice, but it actually turns out to be easier to test this idea in V1, and we did some experiments which I'll just briefly go over at the end to show you how we did that. The reason that we did these in V1 is that you can use this hideous-looking object in V1. This is what's called a Utah array. It is a multi-electrode recording device with 100 different electrodes, and it is like a pincushion. It's very much like a pincushion. And it happens to be advantageous that the primary visual cortex of the macaque monkey, shown here facing you at the back, is largely flat, which means that this array can be inserted into it and can record simultaneously from a large number of cells. And what this allows you to do is to record the, let's say, orientation selectivity of large numbers of cells in V1 at the same time. And so you have a set of data like this, which is in fact a representation of the orientation tuning curves of 70 simultaneously recorded cells. These data were collected by Arnulf Graf, who was a postdoc in the lab. Now you can use the model that Mehrdad Jazayeri wrote to put these data together to estimate the direction, actually the orientation of the stimulus. And you do that using exactly the same schematic system that I showed before. You take these responses of all the V1 neurons, you weight them according to a designed weighting function, and the result is the log of the likelihood that this orientation was present. And you can do that for multiple different orientations. And what you see is that for this pattern of activity, the likelihood is very sharply peaked that the green orientation, which is close to horizontal, was the one that was actually present. And you can capture the performance of the model by saying, if we were to estimate orientation based on these data from this model, we would estimate with high degree of accuracy. We would be right about 65% of the time, and our errors would only be about 5 degrees either side with any frequency. Now, it turns out that correlation, the same correlation that made it possible to detect choice probability, has an impact on how we do this decoding. And we can see that by working through an example where I take this population. Let me just ask about 2 neurons here that have different tuning curves. And what we can do is ask, for just 2 orientations, how do those 2 neurons contribute to the discrimination? So the blue dots show the response of each neuron, neuron 2 and neuron 1, to one orientation. The red dots show the response to another orientation. And the scatter reflects the fact that responses in visual cortex are highly variable. We can scatter these against one another, and what you can see is that there's a line that more or less separates the red from the blue, meaning that appropriately chosen decoder ought to be able to discriminate the red from the blue quite well. The model that we made from Mehrdad's analysis, the Poisson-independent decoder, does that quite well. But you can also see, if you look at these scatter diagrams, that the red and the blue clouds show correlation. That is, the responses co-vary from trial to trial in these neurons. And so what you can actually see under these circumstances is a slightly different line, which can be computed empirically to make a different decoder, which I'll call the empirical decoder. And this last analysis simply shows that the performance of that decoder in blue is substantially superior to the performance of the decoder built from first principles. And if you deprive that empirical decoder of correlation information, its performance collapses again. This is simply another measure of the same thing. So the conclusion I would want to offer you is that the same kinds of circuits and the same kinds of mechanisms that allow cells to capture visual information that's afferent to them from subcortical and early cortical structures and build new kinds of visual representations may be the same kinds of circuits that are actually used to decode aspects of that information. And so when you think about a large network of areas like this, you may want to think about each of these feed-forward connections as relating not only to the computation of new properties of the visual stimulus, but also to the decoding of earlier representations. And let me just close by thanking the people and the organizations that were critically helpful and have honored this work. The work I described was done by Mehrdad Jazairi, Arnold Graf, and Adam Cohn, who were students and postdocs in the lab respectively. The Champalimaud Foundation, of course, is the reason we are here and the reason that you are here, and I thank you all for your attention. And I hand over to my colleague, Bill. Plug the power in before I do that. So you need to fire yourself up.
So how do I turn myself up?
They will turn it on. It should be on.
There we
Go back.
There we go. Thank you, Tony. Wonderful talk. I would also like to echo Tony's thanks to the Champalimaud Foundation and to the jury for selecting us for this prize. It's obviously a fantastic honor in general, and it's an even better honor to share the prize with my longtime friend and colleague, Tony Machen. Like Tony, I also have great memories of ARVO. I remember when I was a young postdoc at the National Eye Institute, at ARVO time, about half the institute would decamp to Sarasota for the week for the ARVO meeting. And I remember these intense scientific discussions on the beach, and I distinctly remember one of these discussions where Bill Geisler reached over into the sand and started drawing data figures in the sand. And pretty soon, 3 or 4 other people were saying, no, no, no. And they started drawing data figures in the sand. I thought to myself, wow, these guys are really hardcore. It was also at ARVO that it was some years later, and we presented an abstract from my lab. A student presented it, and it was a particularly, I thought, clever little piece of science. And Al Ahumada accosted me on the beach later and said, you guys are studying matching behavior. I did my PhD thesis on that in 1959. And these kinds of memories come back. It was at a vision meeting at, I think it was ARVO, where I succeeded Curtis Baker in a talk on motion vision. And Curtis dubbed this random dot pattern that Tony illustrated for you, he dubbed it in his talk Mauve Noise because Tony had done the original programming for that particular version of the stimulus. Fortunately, I had the podium right after Curtis, and I objected publicly to that naming on the grounds of ambiguity because it was only one of many kinds of noise that Tony has introduced into this field. So, as Tony said, the relationship has been like that for a long time. Tony and I were actually driving up, sharing a car with 2 people who didn't know us very well, recently headed up to suburban New York. And one of them observed after about 20 minutes, you 2 sound like an old married couple. So it's with lots of good memories and lots of gratitude that I come to this particular symposium. And I sort of want to take up this talk, take up the story where Tony left off, with a very beautiful correlative studies that he described and the decoding studies showing in principle how MT neurons can provide the information that animals and humans actually use when we see motion and make judgments about motion. And I'd like to take up that story and tell you about some experiments that were done in my lab over the course of about 15 years where we used microstimulation techniques to really try to put that relationship between the physiology and the behavior on a causal basis. And the title of my talk is What Microstimulation Reveals and Does Not Reveal, and that's important, about the neural substrate of motion vision. So here's MT, the star of our story, which Tony has already introduced you to. And the one thing that he didn't mention is that MT neurons are organized in columns. They're direction columns, these direction selective cells. And this is a cartoon, sort of like the Hubel and Weasel cartoon of orientation selective columns in V1, except you'll notice that these arrows are unidirectional. This, like the ice cube model of orientation selectivity in V1, is simplified, and there are really pinwheels and swirls and fracture zones. So this is highly oversimplified. But what we started doing soon after I moved to Stanford, Daniel Salzman, a Stanford medical student, joined my lab. And we said, well, what if we put microelectrodes into the midst of one of these columns, and when a monkey is doing, for example, an up versus down discrimination, and instead of using the electrode to listen to the cells while the monkey does the discrimination, let's use the electrode to electrically stimulate the cells and actually try to insert a signal into the circuitry and see whether that actually causes a predictable change in the animal's behavior. And many of you in this audience know that that's in fact exactly what happened. We did this sort of microstimulation with standard protocols, very small current levels, about 10 microamps, 200 hertz stimulation, sort of trying to get at the maximum that we had ever seen MT neurons respond to in a real stimulus. And we had the monkey doing the direction discrimination task that Tony described to you, where he's judging between the preferred direction of motion and the anti-preferred direction of motion for the particular column that we were stimulated. So we very carefully tailored the discrimination task to the column in which our electrode was residing. And we plotted the psychophysical data like this. This is not a standard psychometric function. It's what we call an unfolded psychometric function, where we sort of define here on the y-axis the measure of behavior as the proportion of preferred decisions. So if we're in an upward column, that would be the proportion of upward decisions. And the coherence of the stimulus is on this unfolded axis, where completely random motion is right here in the middle. And increasing positive numbers means stronger and stronger motion toward the preferred direction, and increasing negative numbers means stronger and stronger motion toward the null or anti-preferred direction. And of course, this monkey's extremely well trained, and he's making virtually 100% of his choices in favor of the preferred direction for strong preferred motion, and almost no choices in favor of the preferred direction at strong null motion. And he's splitting his choices about 50-50 in the middle, as you would expect him to. And we stimulated these monkeys randomly on half the trials, and importantly, we only ever rewarded the animals for giving us the objectively correct answer. So if microstimulation caused him to do something that was incorrect, we certainly didn't reward him for that. In fact, it penalized him. Now, at 0% coherence, there of course is no correct answer, no correct direction of motion, so we would just reward the animal randomly with a probability of 0.5, irrespective of the direction he chose. And this was one of the early results of these experiments. This was a psychometric function we obtained on stimulated trials, and this was a psychometric function on the randomly interleaved, this is the unstimulated trials, and this is the psychometric function on the randomly interleaved stimulated trials. And what you can see is that the microstimulation caused a systematic leftward shift of the psychometric function. In other words, if we were stimulating an up column, the up direction was preferred, we increased the monkey's upward decisions at virtually every stimulus level. And it simply resulted in this nice leftward shift, but maintained the slope of the function, meaning that the monkey's overall sensitivity to the motion signals was unperturbed. And we started getting these kind of results fairly regularly in MT, and in fact, we started occasionally seeing what we called super effects, or whoppers at the time, and motion, the performance on the stimulated trials would be completely pegged up here at 1.0. Now you notice this axis only goes from plus 20 to minus 20, so we just lengthened out the axis from plus 100, which would be over here in the middle of the stage, to minus 100, which would be out here on the curtain somewhere. And Chieko Murasugi, who was a postdoc in the lab at the time, got the world's record microstimulation effect, which is shown here. And this is now plus 100 to minus 100. And you can see that even for 100% correlated dots moving in the null direction, the monkey chose preferred direction on about 35% of those trials. And for these strong motion signals, if you remember back to the movie that Tony showed you, these are very strong motion signals that were easy for you to discriminate, and yet the animal made the preferred direction choice of the column. He made that choice on almost 100% of the trials. So these effects can be extremely strong, and we can quantify these kinds of effects in units of the visual stimulus just by measuring that leftward shift in units down here, in units of coherence. And we did that in initial round in 3 animals, about 100 different stimulation experiments. And this was the population results that we published with Daniel Salzman, Chieko Murasugi, and Ken Britton in 1992. And this simply shows what we call the equivalent visual stimulus. That's the size of that leftward shift. And it's a frequency histogram showing that we got significant stimulation effects on about 60% of the experiments, which are shown in the open bars. In about 40% of the experiments, we got no effect at all. But the important thing is, and there are lots of reasons to get no effect. I mean, the amazing thing here is that this worked at all, right? And so there are lots of reasons to get no effects, mostly having to do with the fact that we were positioning our electrodes blindly in the middle of these columns, and we had no idea whether we were near the edge or near the center or what most of the time. And the important thing is that when we got effects, the 60% of the experiments in which we got effects, we could predict the direction of the change from the physiology of the neurons that we were recording. So we had a few sort of wrong way effects out here. I mean, this is biology after all. But the remarkable thing is that this established a real link between the animal's perceptual judgments of motion and the characteristic of the single neurons and the columnar properties at the end of the electrode. Greg DeAngelis came to my lab fairly soon after this, and Greg and I were impressed by the fact that many cells in MT are tuned for stereoscopic depth in addition to being tuned for motion. This had been observed by John Mansell and David Van Essen and Samir Zeki quite a few years earlier. But what Greg discovered working in my lab was that the disparity selective cells were organized in sort of islands in MT. So MT has all of these elaborate direction columns, but in certain islands, almost all of the neurons would be disparity selective in addition to direction selective. And furthermore, the preferred disparity varied systematically within one of these islands, so that many neurons clumped together preferred near disparities, cross disparities, or signifying near objects, whereas clumps of neurons toward the other end of the island would prefer these far disparities. And we asked whether stimulation of MT could influence direction, the stereo disparity judgments in addition to the direction judgments. So we set up a version of the dots task. If these are two eyes and we're sort of looking down on top where the monkey looked at a screen of random dots, and in the particular part of the screen where the MT receptive field resided, we would actually distribute these dots in depth using red-green anaglyphs. And the monkey's job in this task was to ignore the motion. He's looking at the same stimuli, but he ignores the motion, and he simply tells us, are most of the dots farther away than the fixation point, or most of the dots closer than the fixation point? And of course, simply by varying this binocular correlation, we have a direct analog of the random dot motion stimulus where we can vary the signal-to-noise ratio and get very nice, clean psychometric functions. And when Bruce Cumming came to the lab, Greg and Bruce and I did these microstimulation experiments and showed that indeed we could affect these stereo depth judgments. So this is an example of an MT column that was actually tuned for far disparities, and we stimulated on half the trials, just as with the motion experiments, and we increased the proportion of far decisions, getting a nice leftward shift of the psychometric function. And this is an example of an experiment in which the column of MT neurons was tuned to near disparities, and then we could shift and get more near decisions by the animal. So we could do this, and Greg DeAngelis went on to do a lot more of this kind of experimentation in his lab at Washington University, St. Louis, and now at the University of Rochester. And he's done very, very beautiful experiments looking at the relationship between disparity and motion judgments, as has Bruce Cumming in his lab at the National Eye Institute. Jamie Nichols came to my lab a few years later and undertook a different kind of microstimulation experiment. Many people had asked us over the ensuing decade whether these results were unique to this sort of noisy dot pattern where the monkey's judging one direction of motion or the opposite, and whether it was unique to using eye movements as an operant response. So Jamie tried to answer both of those questions at one fell swoop by doing this experiment. So the monkey looked at a fixation point, and we presented dots in the MT receptive field as usual. But now the monkey was free to report any direction of motion that he saw. We didn't force him to bend his judgments into one direction or the opposite, but he was free to report any direction that he saw. And the way he reported the direction of motion was to manipulate this joystick, which was on a circular track. And he could move the joystick around and around. And as he moved the joystick, this dot would go around and around the aperture. And the monkey just lined up this dot with the direction of motion that he saw on the screen. So he had perfect freedom to choose the direction of motion that he wanted to. And for sake of time, I won't show you the actual data here, but Jamie found that we could get just as good a microstimulation experiments with this sort of veridical judgment of motion and direction. He found a lot of really excellent evidence for a vector averaging kind of mechanism between the actual visual direction of motion and the microstimulated direction of motion. And he showed that we can get this with this output being through the arm movement, and thus it wasn't specific somehow to visual ocular motor integration. So since we did this line of experiments, these techniques have been used to demonstrate similar psychophysical and neuronal relationships in the realm of somatosensory flutter discrimination, with visual memory, Tanya Pasternak and James Bisley, and visual attention, very beautiful experiments by my colleague Tieran Moore, who is now at Stanford, and in face categorization by Hussein Asteki and his group in Iran. And fundamentally, I think that given threshold performance, pushing the system to the edge of performance, and given some sort of organization, columnar organization or topographic organization, this kind of experiment can be done routinely. Now, there's nothing routine about having that precise control of behavior, but I think in principle, these kinds of things can be done for many sorts of visual cognitive phenomena. Now, at the end of the day, we were left with a puzzle that presented to us the limits of what microstimulation can tell us. And I gave a talk here at ARVO in the year 2000, sort of one of these millennial symposia that was going on. And I listed a bunch of key questions about vision that as a central nervous system visual physiologist I'd like to know the answer to. And the one that I listed as most interesting to me was the following. What does the monkey actually see when we stimulate MT? Now, the data that I've shown you are objectively measured data, measurements of behavior, but they're consistent with two different answers to this question. So one possible answer to this question would be that if the monkey could talk to us, he would say to us, hey guys, I saw leftward motion on that trial and I reported leftward motion. Why didn't you give me a reward? But another possibility is that he would say to us, you know, I saw rightward motion on that trial, but I reported leftward. I don't know why the hell I did that. All right? And both of those internal experiences are consistent with the data that I've shown you. And so here we are in the lab just kind of following our noses, doing standard neurophysiological experiments, and we run up against this question, you know, that is really an age-old question that goes back many, many centuries. What are the subjective concomitants of activity at different brain loci? And all of a sudden, here it is in front of us in the lab. And I don't know any way to actually get a rigorous answer to this question without doing this kind of electrical stimulation on human MT. And I've actually floated the idea and discussed with neurosurgeons. What if a volunteer, you know, like me, for example, wanted to have his MT stimulated? Would it be possible? After all, we pay young people to get on top of Saturn V rockets and get shot into orbit. You know, why can't a volunteer have their MT stimulated? That's much, much safer than sitting on a Saturn V rocket. An editor at MIT Technology Review heard me do this. I do it as a thought-provoking exercise for audiences as much as anything else. But she wrote this thing up in MIT Technology Review. Stanford neuroscientist Bill Newsom wants to implant an electrode in his brain to better understand human consciousness. And much to the amazement of people in my laboratory, this got picked up on Slashdot. And those of you who are really hip out there will know Slashdot. People in my lab were much more impressed about that this was Slashdotted than with any publication I've ever published. And Slashdot has this interesting commentary, so readers can write commentary, and some of it makes quite interesting reading. One reader said, this was supposed to be a romantic Valentine's Day present for his girlfriend, complete with remote control. Another one says, the lines between this article and the far side are getting pretty blurry. Another one says, most hardcore scientist ever. He's going to implant it in his own head with no anesthesia. Dude! And this one is a more perceptive observer, and it says the article is full of how he wants to do it, but would probably have trouble getting approval and so on. If this is news, alert the media that one day I want to fly around in a jetpack while robot slaves do all my work and it rains Kool-Aid. There's all these skeptics out there. And this last one was apropos. I wonder how likely it is that he'll wind up with a Darwin Award, right? So it's interesting stuff. And we've had people suggest experiments to us over the years, you know, like when you stimulate and the monkey gets it wrong, does he shake his chair like he's frustrated? And people have suggested that we measure galvanic skin responses and pupil dilation, to try to get at the emotional tone of the animal. And these are all kinds of interesting indirect measurements, but I was never really convinced that they would yield anything terribly insightful. But the last little thing I want to present to you are some recent data from Mike Shadlin's lab, done by Rizbe Kiani and Chris Fetch and Mike himself. And some of you are aware of this. This sort of takes monkey training and monkey psychophysics to a whole new level that I did not think could be achieved. And what these guys did is they trained a monkey on the standard direction discrimination task. So up to this point, everything's normal. And on half the trials, the monkey gets the go signal and he makes the eye movement to indicate his judgment, just as usual. But on half the trials, while the monkey's in this delay period, so this is after he's seen the motion stimulus, and while he's in this delay period, a third alternative target comes on, the so-called sure target. And if the monkey is feeling confident in his judgment, he will ignore this target and he'll go ahead and make his choice and get a large drop of juice if he makes it correctly. But if he's not feeling confident in his judgment, he'll actually choose the sure target and he gets a guaranteed smaller reward no matter what happened on the trial. So this is a form of post-choice wagering, in essence, where the animal can opt for the sure option and always get a reward, or he can gamble that he's really got the right answer and he can go for the larger reward. So this is a way to ask the animal himself, how confident are you about the judgment, the stimulus that you've made and the judgment that you've got sort of formed in your head? And the question, of course, that kind of interests me and that interested Rizbe and Chris and Mike was, on stimulation trials. If you stimulate MT, stimulate MST, how is the monkey going to react to this in terms of the certainty of his judgment? And they have some really nice preliminary data. And this shows the microstimulation data, the control data, and the microstimulation data in the same format that I've already explained to you. And this shows that they can get these microstimulation effects. And this is on trials, the half of the trials that do not have the sure target on them. All right? So this is just normal garden variety MT stimulation performance. Now, this is performance on the half of the trials where the sure target was present, but the monkey ignored the sure target and said, I'm sticking with my original judgment. And you can see we get the same sorts of microstimulation effects, control data and electrical stimulation data. These curves are steeper, and that makes sense, because if the monkey's really certain about these things, he should get higher percent correct, and these unfolded psychometric functions steepen up. So this is nice. This is a good sanity check. The key question, the most interesting question, the sort of titillating one to me, is what happens on trials when he actually opts out and elects the sure target and elects not to take the gamble. And this is the answer. So this is the probability with which the monkey chooses the sure target, with which he opts out against the strength of the motion stimulus. Again, this is strong motion stimulus in the preferred direction, strong motion stimulus in the null direction. And one thing you see straight off is that he has a higher probability of opting out and choosing the sure target for low coherences right here in the middle. And that's logical, right? That's exactly what you would expect. Now, the solid curve here shows the unstimulated half of the data. So that corresponds to the solid blue curve here. And the dashed line shows you the monkey's probability of choosing the sure target on the stimulated trials. And you can see that what happens is that the peak of this curve shifts leftward. So whereas the monkey previously reported being unsure of his judgments very close to 0% coherence, now the peak of this curve is shifted about 10 or 15% dots to the left, meaning that the stimuli with 10 or 15% leftward dots, for example, is the place of maximum uncertainty, which is again consistent with the idea that the microstimulation actually replicates the perceptual effects of dot motion, and the new 0%, if you will, is now 15% opposite of the direction that you're stimulating. Now again, this is not a direct assessment of what the monkey sees. We can't do that without sentient observers who can report verbally what they see. But it is a piece of data that's actually consistent with the idea that MT is really reflecting the motion computations and that the MT stimulation effects are, in essence, just adding dots to the stimulus. So, how can we ever go beyond this? Can we ever do this in humans? Well, what we really need is a way to stimulate the human brain non-invasively, without cutting a hole in the head and without putting an electrode in there. And we need to be able to do that. And the question is, are there any new tools coming along that are imaginable? One that everyone's all the rage about these days is optogenetics. But optogenetics, I think, will go into humans at some point. Carl Diceroth, my colleague at Stanford, is a trained psychiatrist, and his real ambition is to take this technique into humans at some point. But optogenetics is invasive on two counts, right? First of all, you have to do gene manipulations, gene therapy, essentially, to make the cells sensitive to light. And secondly, you have to have a hole in the head in order to get the light pipe in there. I think something that might be down the line a bit, but certainly people are thinking about right now, would be Magneto Genetics. Now, this would be a situation where your channel proteins in the membrane are not gated by light, as in optogenetics, but are gated by magnetic fields. And we know how to focus magnetic fields non-invasively down to very, very fine spatial scales. So the magnetic fields can definitely be focused through the skull non-invasively, but what we need here is real molecular biology engineering to build channels that are magnetically gated rather than voltage or optically gated. A final possibility that many people around the country are thinking about is possibly using focused ultrasound. It turns out when neural tissue is vibrated, cells fire action potentials. And this has been known actually anecdotally for about 50 years in the field, but it's been shown recently in very beautiful experiments by Jamie Tyler and his group down at Arizona State University, and I'd refer you to this paper, the methods paper published in Neurons. The big problem with ultrasound is that the skull does terrible things to it. It reflects it, it absorbs it, it refracts it, it heats up, and ultrasound has gone nowhere in the brain precisely because of this problem. But recently, this Israeli company, Insightec, claims to have solved this problem by doing very fine CT scans of the skull using arrays of ultrasound emitters and being able to basically calculate all of these effects of the skull on ultrasound and back that out of the stimulation. And they claim now, you can go to their website, they claim focused ultrasound down to a range of a few millimeters inside the human brain, and they actually have a machine on the market, and their whole market here that they're aiming for is actually thermal ablation of tumors. And I'm like, wait, let's just back off on that power. Let's don't coagulate anything. Let's just tickle those neurons a bit, and does the subject feel anything? But they don't have IRB approvals for that. They can't do that because of IRB approvals at the moment. But I think, it might be possible to get these things down the line. And if we could do experiments in humans, then I think, all bets are off. I mean, I think a real science of how conscious experiences are linked to physiological activity of certain brain structures might really be possible. And I simply don't think it's possible right now. But it could be down the line, in which case we really would perhaps one day come to the elegant description that Al Sommer provided in his introduction, a single seamless science of seeing. I like that phrase. I want your speechwriter to write for me. That's a very beautiful phrase. Carla Schatz. Carla is my better in many, many ways, but that's a different talk. So I just want to close by saying thanks to the Champalimaud Foundation again. I think this award is important for one reason, because just within its sort of 4 to 5 years of existence so far, they have ranged in granting this award such a broad range from the molecular biology of photon absorption in the retina to the wonderful humanitarian work of organizations that actually do very practical things to make life better for blind people, and now coming into the seam between physiology and perception at very high levels of the brain. And I think Champalimaud, by doing this, has sort of defines this single science of seeing, and I thank them personally for the award, and I thank them on behalf of the field for holding this vision out for us for the future. Thank you very much.
So on that note, let me remind you that you can now begin applying for the 2012 award, which by then €1 million is going to be a lot of dollars. There are applications, nominating forms outside. You can also get them off the web. Please, let's thank Tony and Bill again for great lectures. The jury feels terrific about its selection, and we can join them for a reception out in the hall. So, Tony and Bill, congratulations.